Hello everyone. As we have promised that we are going to be going on to local anesthesia and many other topics of oral maxillofacial surgery. But before we start, there are some concepts about nerve, neuron that we need to be aware of before we go into local anesthesia. Why? Because we can't straight jump into a concept and imagine ourselves to understand it if we don't know the basics behind it. So what I'm going to do is I'm going to introduce a little bit details of neuron, macroscopic level and macroscopic level. So over here we can see, if you are seeing on the screen, there is a neuron over here. Now neuron has is the structural and functional unit of our nervous system. I'm going to briefly introduce. There are about 100 million neurons in our nervous system. Right. Now if we look at this neuron diagram, we'll just slide by. This is a proper neuron with its cells. Now, neuron is not the only cell in our nervous system. We have neuron, we have oligodendrocyte or Schwann cell, we have astrocytes, which are your blood-brain barrier. Then we have microglia, which is nothing but macrophage from blood. So these are several cellular components of your nervous system. Now, if you look at this diagram over here, this is the cell body of the neuron. These nerve endings are known as dendrites. What is the job of dendrites? To receive impulses. To receive impulses. These are nodes of Ranvier. These are oligodendrocytes or Schwann cells, as we have already mentioned over here. Their job is to make myelin sheath. So these are some of the few common questions that are being asked in your MCQ exams as well as sometimes Viva exams in physiology, and it will also help you in post graduation for basic sciences answering all as well. So what makes your myelin sheath? Oligodendrocytes and Schwann cells, which are adherent to your neuron, make myelin sheath. The star-like structure is the cell body with nucleus in the center. What are these branches like? These are known as dendrites. Dendrites' job is to receive electrical impulses. Now over here we can see, if we can appreciate, there is a structure over here, this branch like structure or this stem like structure. This is known as axon hillock. This is known as axon hillock. Why axon hillock is important, we will study in the physiology of the neuron. But just understand, this axon hillock is the site of generation of electric potential. It is the site of generation of impulse. Anyways, this long trunk-like structure is axon, and these are axon terminals over here. These branches over here, which we can see, there are some swollen structures on the ending of every branch. These are known as, I'm sorry, this pen is not working properly. Anyways, yeah, synaptic knobs. So this is essentially a structure of neuron. Again, we'll revise it. This star-like structure is cell body. In the center, neuron, branch like dendrites. Job, receive impulses. axon hillock, which will generate the impulse or transmit the impulse. Then you have an axon through which the impulse gets transmitted in this direction. Then in between we have nodes of Ranvier, which will serve an important purpose in local anesthesia as well as electrophysiology of the neuron. These impulses get transmitted to these axon terminals, and through the synaptic knobs via synapse they go on to the next neuron. So this is a very basic and I will say vague overview of how neuron functions and what are the structures, right? Moving forward, there are different kinds of neuron. This one is known as unipolar neuron. This is known as bipolar neuron. Bipolar neuron, classical example is retina. Then we have something known as pseudo-unipolar neurons. Pseudo-unipolar neurons, which is commonly found in ganglions. Now there are different kinds of nerve fibers. Classically, on the basis of presence or absence of myelin sheath, as we have already seen, myelin sheath is made by something known as oligodendrocytes or Schwann cells. Why there are two different names? This I will cover in a little bit. But for now we will understand that on the basis of presence or absence of myelin sheath, on the basis of thickness, and on the basis of speed of conduction of the impulse, there are four kinds of fibers. Essentially two, in which A and C, but A is further divided into alpha, beta and delta. Now A alpha and A beta I will cover up. First of all, there are A alpha is thickest, A beta is second thickest. A alpha is the thickest with the diameter of about 7 to 16 microns, whereas A beta is 6 to 8 microns. A alpha, because of their thick diameter, can conduct electric impulse at a larger rate, so that is why they are the fastest fibers. So that should automatically make a concept in our head that thicker the neuron, faster the impulse. Then it is a myelinated fibers. Always remember, myelinated fibers are faster than unmyelinated. Why? We will cover. Then A beta, it's the second thickest. 6 to 8 microns is the diameter. 30 to 70 meter per second is the velocity. A alpha's velocity is 70 to 120. A beta is responsible for touch. A alpha is in muscle spindle fibers. These are not that relevant in dentistry or and in oral maxillofacial surgery. For us, important are these the next two. That is A alpha and C fibers. Sometimes this is a viva question as well. That which fibers are responsible for pain. So we'll see. Anyways, A delta, next thick diameter 6 to 8. It is the smallest myelinated nerve fibers with and their velocity is 12 to 30 meter per second. 12 to 30 meter per second. I think, yeah, their speed is 12 to 30 meter per second. Next is they're responsible for temperature and fast pain. What do you mean by fast pain? Acute pain. Acute pain. They are responsible for acute pain or something known as fast pain. Now what is C fibers? C fibers are the smallest fibers. They are unmyelinated. That makes them naturally slowest of the nerve fibers. Their diameter is about 2.5 to 4 microns. Their speed is 0.5 to 2 meter per second, and they are responsible for slow pain, or in other terms we can say chronic level of pain. Chronic pain. So this is extremely important. A alpha and C fibers. This is a very common Viva question, that which fibers are responsible for what kind of pain? A alpha, A for A alpha, A for acute, C for chronic. So we can remember as a slow pain fibers as well, right? So this kind of suffices our macroscopic details. One more detail that I would like to cover is in our nervous system, there are certain terminologies we need to be aware of. So for those terminologies, we need to understand a very simple basic concept that our nervous system is divided into CNS and PNS. C stands for central and P stands for peripheral. Central means essentially two organs, that two and only two organs, brain and spinal cord. Apart from that, any other nerve without any exception in the body becomes automatically a part of PNS. All the nerves, all the neurons, anything related with neurons in these two become a part of CNS, and nerves and neurons which are not in brain and spinal cord automatically become a part of PNS. This concept is very important to understand. Why? I'll tell you. Over here we can see there are a bunch of neurons that I've drawn. These neurons, now imagine this, these neurons, all these are a part of a single nerve. There are many neurons in a nerve, but let's imagine we are seeing a nerve with only these four neurons. Now these neurons will have different kind of cell bodies also. So these cell bodies, if we put them together, they will form like a bunch over here. Let's make a bunch. Let's make a bunch of cell bodies. Now these bunch of cell bodies will look somewhat like this. So this bunch of cell bodies in the peripheral nervous system will be known as ganglion. Ganglion. So what are the most common example of ganglion? Trigeminal, otic. Any other examples that we know of? Short ciliary, the famous submandibular ganglion. Now many of you must be confused that why trigeminal ganglion, or also known as Meckel's ganglion, also known as semilunar ganglion. Why it is when it is almost located in brain. But here is the catch. Trigeminal ganglion is not located inside the brain. It is located in the dural cave. That is, it is in the dural fold. It is not located inside the brain. It is located in the dural fold. It is not part of the brain. That is why trigeminal is a ganglion, and the counterpart of cell body collection cell body collection in central nervous system is known as nucleus. So the famous nucleus ambiguous or nucleus of Tractus solitarius, etc. All these examples are of cell body collection. Same concept, understand it like this. Imagine a neuron in front of you, and imagine 10-20 neurons with their cell bodies forming like a bunch together, and this thing, same bunch of cell bodies in the brain or spinal cord will be called as nucleus. Same cell, and whereas outside the brain and spinal cord they will be called as ganglion. That is the only difference in terminology, that is it. So it is very important to understand that cell body collection inside brain and spinal cord will be called as nucleus, whereas cell body collection outside the brain and spinal cord will be called as ganglion. Similarly, if we look over here, of those neurons, there will be axons coming as well. So those axons will also form a collection. So those collection of axons outside brain and spinal cord, they will be known as the what is the most common and famous name? Nerve. That is the most common and most famous name that we or the layman terms or layman people are also aware of. That is the nerve. But nerve is what? Now we know. Nerve is nothing but a collection of axons outside brain and spinal cord. So next question, what is our question? Next question is, what is this collection of axons named in central nervous system? So this collection of axons in central nervous system is known as tract. Is known as tract. Example, pyramidal tract, or even better, famous example, ascending descending tract of spinal cord. Ascending or descending tract of spinal cord are some of the names of collection of axons inside the brain and spinal cord. So these are the few terms. We'll revise it again. Collection of cell bodies or collection. This is a cyton. So many cytons together in brain spinal cord will be called as nucleus. Outside brain and spinal cord they will be called as ganglion. From those cytons there will be axons coming out as well. So that collection of axons out in the brain and spinal cord will be called as tract. Whereas outside brain and spinal cord they will be known as nerve, which is the most famous name. I hope this much is clear. Now we will move on to the next thing, that is the microscopic structure. That is the microscopic structure. Now we'll come on here. Now in a microscopic structure we need to understand a few basic things. If you look at a nerve, I don't know if this will allow me to draw properly, but we'll see. Okay, yeah, it's allowing me some way. So if we look at this, this is a very rough diagram. I'm trying to draw a diagram which can make people understand what I'm trying to say, but I hope I'm not very good with diagrams actually, but we will try to convey our message as clearly as possible. So nerve structure, it's not just neurons. Neurons need to be protected from the other things as well, such as blood or any other surrounding stimulus. So how do we protect it? So there are four connective tissue layers. The first and the foremost layer is mesoneurium. Mesoneurium. This is mesoneurium. This is mesoneurium. What does mesoneurium do? It is the first of all the most outermost covering. Secondly, it suspends the fascicles. What is the fascicles? I will explain. Fascicles is nothing but a collection of neurons. That is, so what is fascicle? Collection of neurons is known as fascicle. Now over here we will have what we have just studied, fascicles. This is known as fascicles. There will be small fascicles, there will be large fascicles of any shape, size, whatever it is. So these are known as fascicles. These fascicles will have small neurons in them. This is an example that I'm drawing. We'll understand right now. Now from outside to inside if we go, this is a fascicle, right? Now this fascicle in turn will be covered with another layer that is continuous with the mesoneurium but inside it, and that is known as epineurium. That is known as epineurium. And just like that, it covers all the fascicles. Then inside epineurium is the perineurium, and inside perineurium is your last layer endoneurium, which covers the individual nerve fibers. So what are the four layers? First is the foremost is mesoneurium. Inside mesoneurium is epineurium. Inside epineurium is perineurium, and perineurium's inside is endoneurium, which covers the nerve, which we have just studied in the cyton structure right now. Now we will see individual connective tissue layers function. So first is mesoneurium. What is mesoneurium's job? Suspending the nerve trunk. This, yeah, we have seen it is suspending the nerve trunk. Its blood supply is segmental. What is segmental blood supply? So let's see over here. We will try to draw a nerve diagram. This is a cross section. So it has a segmental blood supply. So each segment will be supplied by a different source of capillary from the same artery or from the different arteries. So that is known as segmental blood supply. It is continuous with the epineurium. So the thing is, over here if we see, this mesoneurium gets mixed up with your epineurium and it covers all the fascicles. Now we'll go on to epineurium. What is epineurium? It is the second connective tissue layer, as we know, from outside to inside. What was the first? Meso, then epi. It is a loose layer. Its function is to protect from the mechanical stresses. This is a very important question. What is the function of epineurium? Protection from the mechanical stresses. It has longitudinal collagen fibers. Now what is longitudinal collagen fibers? The thing is, what we are seeing right now is a transverse section of nerve. Transverse section means I'll take this nerve and cut it like this and then see it this from this angle. But longitudinal section means in this way. So if we take up epineurium layer, so what are we going to see is this will be the epineurium covering, these will be the fascicles, and we can see a large number of collagen fibrils coming like this. And since they are arranged along the nerve, that is why they are known as longitudinal. I hope it is clear. Now epineurium consists of two components. One is epifascicular layer, another is interfascicular layer. What is epifascicular? Epifascicular means which is outside the fascicle. Simple. There is no tough concept in this. This is a fascicle. Anything out the epineurium outside the fascicle is epifascicular. Whereas, let us say there are multiple fascicles over here. So whatever epineurium that will be found in between these fascicles is known as interfascicular layer. Simple. Nothing, no tough concept as of now. Epifascicular outside, interfascicular between. That's it. Now, what is the clinical aspect of this? The clinical aspect is when the nerve trunk gets compressed, or let's say we are excessive dissection during surgery. Sometimes if whatever edema or inflammation happens, it happens in the epineurium layer only. So does it happen in meso? No. Does it happen in epi? Yes. So where inflammation happens or where inflammation starts, it starts from epineurial, which is known as epineurial edema, causing in turn something known as epineurial fibrosis. Epineurial fibrosis. Epineurium also contains of lymphatic channels. Epineurium also consists of vessels as well, and those vessels are famously known as vasa nervosum. Vasa nervosum. So vasa nervosum, what they do is they'll be like these small capillaries again traveling longitudinally in the epineurial layer. This is known as vasa nervosum. Again, as we see, longitudinal course, just like how I showed right now in this diagram. And what these vasa nervosum do is, I'll explain. Let's say this is a fascicle. Okay, maybe this is a fascicle actually. Better if we draw like this. This is a fascicle. These are some individual nerve trunks where you have perineurium surrounding them. We'll draw perineurium with a different pen actually to make it understand. Yeah. So we have perineurium surrounding different nerve trunks. Within them will be your endoneurium surrounding different nerve fibers. Over them is your epineurium. Right. Now understand something over here. In the epineurium, these are, let's say, this vasa nervosum. So vasa nervosum's only job is not to supply epineurium. Vasa nervosum, while they travel along with the epineurium, they send out small channels inside, small channels, which penetrate the perineurium and directly go to endoneurium. Penetrate the peri and go to endoneurium, and they supply endoneurium of your nerve and in turn responsible for nerve supply, in turn responsible for vascularity of individual nerve fibers. So from supplying a proper nerve trunk to fascicle to nerve individual nerve fibers, vasa nervosum is responsible. What is responsible? Vasa nervosum. How? It will travel along the peri, it will travel along the epineurium, I'm sorry, penetrate the perineurium and go to endo and supply. Go to endo and supply. Simple. No difficult concept as of now. Then let us move forward. So over here, it penetrates the perineurium and connects to endoneurial small vessels or endoneurial capillaries. Endoneurium itself will have small capillaries, and it will communicate with these vasa nervosum, and they maintain the vascularity of a nerve. They are responsible for vascularity of individual nerve. Moving on to the next point, we have perineurium, the third layer. Perineurium is a very important layer for the point of view of oral and maxillofacial surgeon. Why? Because perineurium poses as the greatest barrier to LA. Poses as the greatest barrier to LA. This is a very important bio question. This is a very important MCQ. This is a very important bio question for postgraduate level as well. Whenever we present seminars or JCs, which layer prevents LA entry the most? It is perineurium. And in perineurium also, which one? Something known as perilemma or the inner cellular layer. Or the inner cellular layer. Again, bio question. Let's move on. So perineurium is nothing but a pia mater continuation. Is nothing but a pia mater continuation. What is pia mater? Brain, as we all know, is covered with three layers. Can be remembered as DAP, Dura, arachnoid, pia. So perineurium is nothing but a pia matter continuation. Some people say it as pia arachnoid continuation, whichever way you want to understand. It surrounds endoneurium sheaths. It has two layers. One is outer, second is inner. Now, this is a very important point. Another MCQ question, another Viva question. What is responsible for thickness of a nerve? Perineurium. Perineurium is responsible for nerve thickness. More perineurium layers, or let's say epithelial layers, more will be your diameter. So perineurium composed of outer dense layer, which is a connective tissue layer, and inner stratified squamous epithelium. So as there will be more stratified, more squamous cells over one another, this will lead to thick perineurium. This will lead to thick neuron instead. In turn, sorry, it will lead to a thicker neuron in turn. Right? Now within perineurium, you have your fascicles, which cover your fascicles. Now these fascicles, now the number of fascicles decide what kind of nerve fiber it is. Now a nerve fiber can have a single fascicle, which will be very large, with concentric layers of perineurium, epineurium, or it can have oligo. What is oligo? 2 to 10 fascicles with perineurium and epi and interfascicular neurium as well. Here, there is no need of interfascicular. Interfascicular is absent in monofascicular. And what is polyfascicular? Polyfascicular means more than 10 fascicles. Now, what is the clinical relevance of fascicular pattern? Why do we need to know the number of fascicles? Because once again, this is a very important detail for OMFS, that whenever you do nerve grafting, fascicular matching is very important, and that is why sural nerve is considered to be a very important and suitable nerve graft in case of injury to inferior alveolar or lingual nerve. It is more suitable for lingual, but for injury to inferior alveolar or lingual nerve, sural nerve is considered to be a more important graft because the fascicular pattern matches for them. And this is another important question. This is another important question. So why only sural nerve is taken? This is a question that you have been asked, because the fascicular pattern matches. And what is the fascicular pattern? This is the fascicular pattern. Number of fascicle is means fascicular pattern. Simple. One fascicle mono, 2 to 10 oligo, more than 10 poly, which is an example of lingual and inferior alveolar. So sural is a polyfascicular, lingual and inferior polyfascicular, and consists almost the same number of fascicles, which we will cover in microneurosurgery lecture later on. And last but not the least, we have endoneurium. We have the endoneurium, which surrounds individual nerve fiber and Schwann cells. So you will be having nerves. This will be your Schwann cell. Let's say make it one more. And there will be a single endoneurium surrounding them. Something like that. So endoneurium's clinical application we'll discuss. It has endoneurium plus endoneurium capillaries. Now if you remember, in epineurium we discussed this very clearly. In epineurium there was something known as vasa nervosum, which was penetrating peri and supplying endo. Now these are the same endocapillaries which are receiving supply from vasa nervosum. So them along with endoneurial, if they ever get damaged, again they will cause fibrosis, and they form solid fibrotic fascicles. They form something, these fascicles which we have discussed just now, these fascicles because of endoneurial damage will undergo fibrosis, and they become solid in nature. They become solid in nature, and due to this solid nature, their regeneration becomes very slow. And this is one of the most famous concept and most famous reasons why nerve regeneration is slow. So people have been saying it from a very long while ki why nerve regeneration is absent or slow. It itself is a controversy. It is slow, it is not absent. We have, in school life we have heard that nerve does not regenerate at all. It's not like that. It regenerates, but at a very slow pace. But why it generates at a very slow pace? Because of endoneurial damage. Because of endoneurial damage, which causes endoneurial fibrosis, and this fibrosis causes fascicular fibrosis, which slows regeneration, which slows down regeneration, or some people say stop regeneration. This concludes our introduction to nerve microscopics and macroscopics. We will upload a lecture soon on a brief overview of cranial nerves. Stay tuned, stay hungry, stay humble, be curious, keep learning. Thank you.